Hello, good morning, and welcome to this week's seminar. We have the pleasure to have with us Dr. Colleen Doherty from the Department of Molecular and Structural Biochemistry at our university here, North Carolina State University. Dr. Doherty's lab investigates the connection between time and stress in plants. Two major objectives is first to use time as a tool to interrogate the signal networks that allow a plant to perceive and respond to stress. And second, the lab is very interested in understanding how changes in temporal patterns affect the productivity of crop species. Colleen is responsible for the photo that we use to announce this seminar series, and a few people guessed it very right. Yes, it's pokeweed, it's Phytolaica americana. Colleen does some very interesting work with that species. Her work, I think, has a lot of potential implications for breeders. Colleen, thank you so much for accepting the invitation to present today. I'm going to stop sharing and give you the floor to you.
Thank you very much for the opportunity to present today. I'm very excited about this. As I know absolutely nothing about plant breeding, and I think it's going to be great to get to talk about this work and get some potential for connections with people in this area. So today I want to talk a little bit about what my lab has done for the last 8 years now, and also focus in on what we're doing and where we want to go. So but I really want to talk about this Plants as Climate Solutions part of my lab, which is fairly new. So what I'm going to do is give a brief overview of these two stories. Both of these are published, so I'm very happy to talk more about them if people have questions, but I'm just going to try to hit the high level points so that I have a little bit of time to just introduce Plants as Climate Solutions. So when I started my research career, I was really interested in how plants make decisions. So I wanted to ask, how do plants, when they're faced with a choice, they have an environmental change, such as increase in temperature, how do they allocate their resources? Do they make a decision to continue to grow? Do they make a decision to reallocate their metabolism to provide a change to that, say, example, change in temperature, to change their metabolism, produce secondary metabolites? Or do they just give up and say, I hope the next generation does better, and go on and make the decision to just go forward with reproducing and try and get as many seeds out as possible? And so what's really exciting about plants is all of this is hard-coded into the genome. And so theoretically, we should be able to take a plant and examine, how does it give it the stress, and examine how it makes those responses, and look at the biochemical changes and elucidate that, and work backwards to try and understand, how those decision-making processes are encoded. What are the biochemical switches that allow it to make those decisions? And so this is complicated by the fact that the plant isn't just dealing with a sudden change in temperature, it's also dealing with biotic stress, other abiotic stresses, nutritional stress, different developmental stages, different times of year, different times of day. And so it's really got this super dynamic background that it's dealing with these changes in. And so it almost becomes like this really complicated problem. And the question is kind of, how do plants make a good decision in an almost unpredictable world? Because there's so many features going on. And by good decision, what's an evolutionary successful decision? So how is that selected for? Because the time they get the temperature change now might be very different than when their offspring get the same temperature change. Maybe they have pathogens now and their offspring don't have pathogens at the same time. So how is, this dynamic world, how are these responses encoded in that background? And the key here is almost unpredictable, because there are some things that are very, very predictable. You all have to pay your taxes on April 15th, and that's very predictable, unless you're presidential or something like that. But most people have to pay their taxes on April 15th. But there are a few other things that are really predictable. So the sun will rise in the east every morning. If it doesn't, you won't have to worry about paying your taxes. Also, the Earth rotates around the sun with the same tilt every year at approximately the same pace. And so those two fundamental physical activities result in a plethora of biological and physical patterns that we see over and over again. So we can see yearly patterns of temperature, daily patterns of temperature, daily and yearly patterns of light. And these manifest in the form of biological patterns. So we see patterns for pathogens, or pests, patterns for beneficial insects. We see patterns of senescence. And for those of us with allergies, we see patterns of allergies. And so there's cycles and cycles and cycles within cycles. And all of these are really fundamentally integrated in loops to these two fundamental processes, the Earth rotating around its axis and then rotating around the sun. And so plants are not stupid, and they have, through selection, realized that if they take advantage of these recurring patterns and these recurring themes, that they can maximize their responses. And so through evolution and selection, the plants that we still have with us are those that were able to utilize these recurring patterns to make these decisions and to optimize their biochemical activities. And so essentially what I like to frame it as is that when a plant's faced with a decision, it really incorporates time management into that decision. So like all good leadership people were taught, you need to manage your time and you need to think about time in the decisions and the time of your lab members. And so plants are really good at this. They incorporate time, and they do this through their circadian clock. So if you're going to be good at time management, you have to know how to keep track of time and not go over on your talk when you're trying to fit in too many things into a talk. So plants do this through their circadian clock, and the circadian clock is a biochemical signal integrator. So it's a bunch of molecular components, it's a bunch of proteins and transcripts that take inputs from the environment. So these could be light cycles, temperature cycles. In the case of us, it's our eating cycles. And they integrate them so that the internal biochemistry of the clock is coordinated with the external environmental changes, these recurring patterns that I talked about. So the plant will photosynthesize during the day when the sun is out, and it's actually going to start that before the sun even rises up so that it's anticipating that and it's ready to catch the first rays of light, and it degrades starch at night. It's prepared for pathogens when they're going to be there, and it's not prepared when they're not going to be there. So, and we know that the plant integrates these signals because we can do experiments where we can jet lag the plant. So if we take a plant, we grow it in the chamber on Raleigh time, and we fly it to the chamber right next to it, which is set on Japan time, and give it a 12-hour time difference, just like us, the plant will get jet lagged. But just like us, it will eventually recover and be set to Japan time, and it'll start photosynthesizing, at before dawn Japan time. And if we fly it back to the chamber next door, back to Raleigh, it will go through jet lag in reverse the same way we would, and it will have to readapt. But we know it can adapt, and that fine tuning between these, environment is what the clock does. It helps ensure that those internal biochemical processes are properly timed to these external activities, and it keeps track of that across changing cycles. And as a consequence, almost all activities that the plant does, anything, I challenge you to think of something that isn't regulated by the circadian clock. I still hedge my bets here and say almost all, because I don't know, there may be something out there that isn't circadian regulated, but I haven't found it yet. And this is true across many different plants. Maybe if we go look up at lichen way up at the Arctic Circle, maybe they are, but they're not exactly pure plants anyway. So most activities are regulated by the circadian clock. And so this regulation occurs through many different levels. So we generally focus on transcription because it's the easiest to measure, and we could get the range of all the transcripts in the plant. And so this example here is from rice. And so this is, each row here is a transcript. And this is all of the transcripts that we could detect in field-grown rice plants. And these columns are different times of day. And so these are organized by their peak expression as shown in red. And so this group of genes peaks in expression right at dawn, these ones a few hours later, these ones in the middle of the day, these ones at dusk, et cetera, et cetera. And so you can look at this and you can see there's this really tight organization of groups of genes that behave coordinately. And it's just a beautiful wave going down at the time points we sampled. And we can see these really nice patterns. The other point of this is that if you look at any one of these time points and you think about you're going to do some experiment, you're going to measure some trait, that landscape you're measuring on top of is different. At this time point, the transcriptional profile is different than at this time point, than at this time point. And that's for transcripts, but that holds true for proteins, that holds true for metabolites, and that holds true for physiology. At sometimes a day, the plant is allocating its resources to that sink tissue and filling up that pumpkin, which looks kind of like a balloon. And at other times a day, it's collecting that carbon, assimilating that carbon. So really, we see kind of global control of biochemistry by the circadian clock. And of course, this affects stress responses because I showed you all of those transcripts are affected. Stress responses are in those transcripts. And so this is kind of really the reason I got into this, because if we want to study how a stress response occurs, we have to understand the clock. And so when I was in graduate school, I was working on low temperature tolerance, and it was all of my experiments were ruined because of the time of the day or different circadian components. And so I just decided if I'm going to understand how stress response works, I have to understand how the clock feeds into this. And so probably the best studied example of this is in herbivory. So if we look at this worm called Teeny, and you place it on Arabidopsis rosettes. The Arabidopsis can produce some chemicals and kind of fight it off. The worm takes a few bites, but it doesn't kill the plant. However, if you grow the plants in day-night cycles and you grow the worm in inverted cycles, so when the plant thinks it's night, the worm thinks it's day, and you place them on there, the plants get decimated. The worm eats up everything. The plants don't produce any comeback. They're just dead. And so this is called circadian gating. And so what we see is that what that means is that at one time of day, at some time, the plant can actually mount a response, and at another time of day, it can't. And so this circadian gating happens for a lot of different activities. It happens for photosynthesis. So if you shine a light on the plant at night, it doesn't immediately start photosynthesizing it. It not expecting the sun to come out at night. And so a number of activities are gated and many stress responses are. And so by gating, we know that the same exact stimulus on the same exact genotype, even on the same exact plant, can produce a very robust response at one time of day and no response at all, or a minimum response at another time of day. So we have a permissive time and an inhibitory time. And that could be because at this time of day, the plant can sense it, or it can propagate the signal, or it can manifest the response. Anywhere along this pipeline could be affected. But what's really cool about circadian gated phenotypes is that now we can take the exact same genotype, we can take the exact same stress, and we can ask what's different about time A and time B that lets this response happen here and not here. So what's the difference between those two? And so that permissive versus inhibitory time can really be a good tool. And this is where time as a perturbogen or a changing component can really help us elucidate these pathways. So that's one of the things I'm super interested in, is can we use time to elucidate these pathways? And so we're interested in temperature stress. And so we know that plants usually expect to have warmer days and cooler nights. We all do. Insects do, we do, plants do. And so we wondered, is heat stress response gated by the circadian clock? Does this show a robust stress response and a inhibitory stress response? And so I have to explain a little bit about how we do our heat stress response. These experiments are all done in Arabidopsis that I'm going to show you here. And so what we do is we take Arabidopsis in its natural growth condition of a perfectly incubated 22-degree chamber, and we move it suddenly to a high temperature. And so if you do that, you take it suddenly out of its happy 22-degree chamber to a high temperature. Generally, depending on the age and the growth conditions, it can survive up to about 42 degrees. If we first give it a non-lethal temperature, this is known as acclimation, it can then make changes. It induces a transcriptional pathway, and it can survive to a higher temperature than its basal tolerance level. And so this is known as acquired thermotolerance or acclimation. And what's really cool in Arabidopsis is these pathways are very well known. We know the transcription factors. It's mostly transcriptional regulation that controls this through the heat shock factors and their subsequent pathways. And we know these pathways really well. And so what we wanted to ask is, well, does the time of day play a role in this? And so we did a very simple experiment. Well, simple for me because I didn't have to do the experiment, not so simple for the undergrad and graduate students that had to do the experiment. But the idea was they would treat the plants at dawn, midday, dusk, and midnight with either that basal heat shock where they just took them out of their happy 22-degree temp chamber and gave them, either a 40-degree, 42-degree, et cetera, looking at these different temperatures and see what temperature they die at. Or we first acclimated them at 38 degrees for an hour and then moved them to these temperatures and see what they died at. And the question was, do we see differences in either that basal tolerance or the acquired tolerance, depending on the time of the day we do these experiments? So theoretically, a simple experiment, actually pretty challenging to implement because it takes a lot of plants. And so I'm showing this example here. So this is sometimes a little hard to see some of the green colors, but I'll walk through it. And then this is the corresponding graph for many replicates of this experiment. So if we look at dawn, midday, dusk, and midnight, and then this is without acclimation. So this is that basal tolerance, and this is with acclimation. And then you can kind of see the green level where it starts to peter off here. So at dawn, they're actually the most sensitive, and that corresponds to the graph here. Their basal level is the lowest at dawn across many experiments. We've repeated this several times. Midday and midnight actually show very similar levels of tolerance, both basal and acclimation. So those show very similar. And surprisingly to me, dusk showed a very robust acclimation response with a super high acclimation response. This is actually higher than most people report. And I made the undergrad do this many, many times because I did not believe them that it could be this high. And then I had a graduate student do it. And then I had another graduate student do it. So this is a really high level of tolerance we're seeing at dusk. And most people just don't make their graduate students come in the middle of the night. So we see, these higher temperatures, I think, because of the time of day we're looking. So the answer to the initial question is yes, heat stress response is gated both at the basal level, dawn tends to be the lowest, and at the acclimation where we see this really robust acclimation in the PM samples, in the dusk samples. And so now we wanted to go back and say, well, we know all these heat shock factors. We wanted to look and say, we know this is acclimation is mostly transcriptional regulated. Can we kind of connect these dots between this difference in physiology and the difference in transcription? So another simple experiment, this one actually was a little bit simpler. And so we just took the plants out of the chamber at dawn and we gave them 30 degrees for an hour, or dusk and we gave them 30 degrees for an hour. And here, we chose 30 degrees because that is the lowest temperature that's been published that they can actually induce that acclimation response. So we wanted to get to the bare minimum kind of level there. And so when we do that, we see kind of what I thought we would see a lot of is these time of day independent transcriptional responses. So what these graphs are showing, this is the control for morning, control for evening, and then heat stress in the morning and heat stress in the evening. And so this gene, which is a known transcriptional regulator involved, master regulator of heat stress response, shows a very robust response in the morning and a very robust response in the evening. So it's time of day independent. We call it no effect for time. And likewise, we can see genes that show time of day independence for being downregulated in response to heat stress. But what was surprising to me was that this was a very small portion of the genome, of the transcriptome. So only about 22% of the genes showed this no effect response. We did see a lot of these genes that showed this gating response, where they had a very robust response at one time of day. This gene goes up about 1,000-fold in the morning. It is actually induced at night, but it's so small compared to the morning. This brings up another question, is it just getting turned on that matters, or is it the absolute level of the transcripts that matters? And that opens up a whole nother can of worms about, which is actually important. But if we just look at like the absolute level, there's a substantial difference between the induction in the morning and the evening. But what's surprising about this is that we actually see a lot of these classic heat shock genes showing this time of day gating. They're very responsive at one time of day, which is the morning, and not at the other time of day, which is the evening. Even though they are responding, they're not as. And what's surprising to me about this is the plants are actually better at acclimating in the evening. So that's like contrary to what we're seeing here. And so the first thought we had, which was probably like, this can't be right, is maybe we're just biased because we always do our experiments in the morning and we see this kind of response. But that idea was reinforced when we started looking at the genes I was really interested in, which is these night responsive genes. So we were really interested in the genes that are coming up only at night and showing that nighttime response. And I was like, okay, we're going to get these genes. They're going to be transcription factors. We're going to test them for their heat tolerance. Most of these genes are genes of unknown functions, genes related to genes of unknown functions. You know, there wasn't a lot of help there for what these genes were doing. So these night specific transcripts generally don't have previous association with heat stress. They're not the classic heat stress response pathway, even though the plant is acclimating better at night. So if we look at those two, like gating only and the no effect only, that's about 60% of the transcriptome. So what's going on with the rest of them? Well, the rest of them has to do with that fact that I mentioned that the transcriptional landscape really varies depending on the time of day you do your experiment. And so if you have a gene that, you know, if the gene is cycling and has a peak of expression and a nadir of expression, if the gene can't be induced more than it is at its peak, and you do your heat stress here, you won't see anything. So you'll see this is an example here. This gene's highly expressed in the morning. It doesn't go up with heat stress. So if you were doing your experiment in the morning, you would think not an interesting gene for heat stress. However, if you did your experiment at night, when this gene's at a lower point, you can now see that it's actually induced in response to heat stress. And so if we look at these genes that are showing this response at night that is masked, we call it masking, is masked by the basal level in the morning. And we look at we think that this is a missed opportunity for regulation of these genes because we've never looked at them because nobody comes in the middle of the night to do these experiments. And so if we look at those promoters of those genes and ask is there anything in common in the promoter of these genes that are either gated to be responded at night or masked, and we see the response at night, we find novel cis-regulatory elements enriched for these. And so these are candidates for novel heat induction pathways that may have been missed before because of the time people do their experiments. Likewise, we can envision if your gene is downregulated by heat stress and it's already at its lowest level in the morning, you might miss it as being downregulated because it's already off. It can't go more than off. And so you would only see it if you look at a time when it's on. And so this is just really doing 2 times, and we can see, you know, genes that we would miss just based on their basal level. And so we call these, you know, masked genes or basal level genes. And so they're about 12% of the genes that have that kind of masking effect. They're either already at their potentially highest level in the 2 time points we have, or they're already at their lowest level and you can't see them getting shut off. And then we have a large group of genes that are kind of a combination of gating and the basal level. So either they're higher in the morning and then they go up more and they're not responding at all at night. And so these are super interesting. So what is it? You know, now we have these candidate genes. So what mechanism is allowing this gene to respond only at this time of day and not at this time of day? Or this gene to respond only at this time of day and not at this time of day? And so we can start asking what factors are involved in that. Are there chromatin differences there that are enabling that? But the key point that I really want to emphasize from this part of the talk is that if you look, about 75% of the transcriptome is really dependent on the time of day you do your experiment. And so this means it's dependent on your clock. And most of us are diurnal creatures. And so I think we generally have an inherent bias in all of our experiments because we have to go pick up kids from school and we have, you know, other lives and we have social clocks that make us do our experiments during the day. And we miss a lot of these, particularly for transcript and molecular work, a lot of the things that are happening at night. So in summary for this section, each temporal perspective is unique. We picked 2 time points, but I think if we picked midday, we would see a different perspective. And so no matter what time of day you do your experiment, there's going to miss some and gain some. There's probably no perfect time, but knowing that there's more than one network response. And we have this classic heat stress response, which is the heat stress response that happens in the morning, but there's another heat stress response that happens at night, and that is not the same. And so there's different networks depending on the time of day. And then the range of responses is a trait that I think we should think about investigating. So for heat stress, we see a difference depending on the species, depending on the genotype, but how much they vary between dawn and dusk. Some don't vary very much and some vary a lot. And so that range might indicate a trait for robustness that I would like to explore further. And then the last thing is just this life at night is something we're missing. We don't see these, you know, night responsive genes are largely unknown. The cis regulatory elements are largely unknown, even for this well-studied example, such as heat stress. And so experiments that we do are really biased by our own internal clock. So that's, the time and temperature. And then the next topic I'd like to talk about is how climate change is kind of impacting that, and how thinking about through the lens of time is affecting our understanding of climate change and making it pretty depressing. So I hope I convinced you in the introduction that plants really utilize time to properly time their stress responses. They vary their stress responses depending on the time of day. It's well documented for biotic stress. It's becoming more documented for abiotic stress. The problem is that things that the plant uses to set its time is changing. And so what we see, I'm just showing a few examples here, but we see poleward movement of both plants and pests and beneficial insects. We see earlier springs, and both of these affect the intersection between photoperiod and temperature. And that's really the heart of the clock, is connecting photoperiod and temperature. And so now plants are seeing 80-degree days when the day length is 6 hours, and that's something they've never seen historically before. And so that's really disrupting kind of these mechanisms for setting the clock. And if we're disrupting setting the clock, we're probably going to disrupt the plant responses to stress, and those stresses are getting worse. So this compounding problem. And then one of the stresses I'm really interested in, one of these pattern changes we're seeing, is this asymmetric warming of daily temperature cycles. So I've mentioned plants expect it to be a warm day and a cool night, and they expect that delta to be very consistent, depending on where you are. In deserts, it's not as consistent, but in most crop growing areas, the delta between day and night is fairly consistent during those seasons. And it's true for North Carolina. And so what's happening is that nights are getting warmer faster than days. And this has a role for the clock. And I'm going to go through a little bit explaining that. So I mentioned if we take that plant from North Carolina and we fly it to the chamber in Japan, which is right next door, we can see that it'll adjust. And it does this through different mechanisms. So if we grow a plant in bright days and dark nights, as indicated here by the yellow is the bright day, the black's the dark night, it will have, this is a gene showing a circadian pattern, a rhythmic pattern. If we release it to constant condition, it will maintain that pattern. So we know that photocycles can entrain these genes. And if we look across the whole transcriptome, about 60% of the genome cycles in these. Some of that could be clock, some of that could be the light regulating it. And in constant light, about 30% continues to cycle. So we know photocycles are important for entraining these. But plants can also use thermocycles to set their clock. So if we grow a plant in constant light, but we give it warm days and cool nights, we can actually set it up with that cycle and then release it to constant light and constant temperature, and 30% of the genome will continue to cycle. It's not the same 30% that cycles in response to the photocycles. Some of them overlap. Some of the same genes are in both, but they have different patterns. And we can do kind of more natural conditions. I do natural in quotes because these are all done with square waves where the light and the temperature come on, they stay on and they go off. But natural, for a molecular biologist, natural. And so if we combine photocycles and thermocycles, and then we release the plant to constant light and temperature, we still see about 30% of the genome cycle, but it's not the same 30% as in either of these. So there's some emergent behaviors that come out. Sometimes photocycles dominate, sometimes thermocycles dominate, sometimes one won't cycle in either of these and then shows up when you have them both. So there's some really cool stuff going on between the intersection between photocycle sensing and thermocycle sensing, but we know very little about how plants sense thermocycles. This is an example of a gene in rice. So here in photocycles, it has this pattern of expression. In thermocycles, it has this pattern of expression. If you combine them both, it has this pattern, which looks closer to the thermocycle. So this would be one where we'd kind of say the thermocycle is kind of dominant when you have them both, but it's almost a mix of the two. And we've also done a few physiological experiments to ask, how thermocycles are playing a role. And so if we grow plants in a 10-degree day-night temperature difference, a 5-degree day-night temperature difference, or a 3-degree day-night temperature difference, we see in that 10-degree, they grow a lot slower than either of the other two, and they're more tolerant to heat stress, to salt stress, and to reactive oxygen stress, where we just add a reactive oxygen generator. And so what we kind of call it is like the slow and steady. With the thermal cycles, they're more robust, they're going a little slower, but they're more responsive to these stresses. So this is, all of this is just to convince you that thermocycles are important. We know very little about how plants sense these thermocycles and how they integrate them and what the function of those genes that are regulated by thermocycles are. And to me, that's really concerning because what we see with warmer nighttime temperatures and with climate change is that those thermocycles are shrinking. And so we know that temperatures are rising, but what's happening is that while both the daily maximum temperature and the daily minimum temperature are rising, the minimum is outpacing the maximum. And so the delta is shrinking. And so basically nights are getting warmer faster than days. And so the amplitude of the day-night temperature difference is shrinking. And we know these warmer nights have significant impacts on crops. Rice has kind of become this model for warm nighttime temperature, although wheat is kind of catching up. And really we see a decrease in both yield quality and quantity with warmer nighttime temperatures. So I was really interested in asking, do these temperature cycles and the role of the clock have any contribution to either the yield loss or the grain quality changes? And so I worked with Dr. Krishna Jagadish, who was at Erie at the time in the Philippines and is now at Texas Tech. And so he had these field chambers here. These are ceramic heaters. And on the inside of the heater, we heated the plants 2 to 2.5 degrees only at night between dawn and dusk. And in the day, they were the same as the control plants here planted next to them. And so this is the temperature from that. It looks mostly blue because the day temperature was the same, and that's the control is in blue. And you can really only see the red peeking out at night. And so this is just a subset of that. And I want to emphasize that, during the day, there's no difference. And at night, they're not getting a heat shock ever. They're just not going down as much as the normal nighttime temperature. And they're maintaining that 2 and 2.5 degree difference. And I want to point out that, the delta between day and night is much bigger than the difference between the normal nighttime temperature and the warm nighttime temperature plants. So this is a very small, subtle change, but like has been previously shown by several groups, we see a decrease in yield even with that small change. So we see about an 8% decrease in yield. And so we wanted to ask, does the clock have an effect? So we sampled every 3 hours throughout the circadian time points, and we wanted to investigate, how is the clock having an effect? And we looked at a number of things, but I'm just going to talk about the transcript stuff, which is published. So here we looked at, just to come back to the point that you really need to look at multiple time points, and a single time point isn't enough. If we just look at differentially expressed genes, we see that, this is just upregulated genes, so genes that are induced in response to high nighttime temperature. No single time point captures them all. These are genes that are uniquely responsive at only this time, uniquely responsive at only this time, uniquely responsive at only this time. So you really need to sample multiple time points to capture the full picture. And also by sampling multiple time points, we can look for some of these interesting patterns. And I'm going to highlight 2 patterns that I'm pretty excited about that we see, because we're interested in the regulators of these patterns. So one of these, is kind of best represented by this light harvesting complex B, which is a known photosynthetic component. And like all good photosynthetic components, it's anticipating, its transcript level is anticipating dawn here in the control conditions. It's coming up. And then as soon as the light hits, the clock and the light work together through ellipse and really induce that to a very high level of expression. And then it maintains that throughout the day. And then before you even get dusk, it's already coming down at the transcriptional level and shutting off. So this is classic photosynthetic regulation of transcripts. In warmer nighttime temperatures, I kind of make the analogy that these behave like teenagers. They're waking up late, they're groggy, they're not reaching their maximum at 3.5 hours. They're waiting to 7 hours, and then they're trying to overcompensate. They're coming up to a much higher level, and then they're going off at the same time. So we see this kind of delay in their activity. And so this is light harvesting complex B. If we look across all of the transcripts in all the transcripts that we could detect, this is the figure I showed earlier from the control. They're very neatly organized. We can see groups of genes and this kind of nice wave of expression. Under warm nighttime temperature, that wave is really disrupted. The genes that are cohort, that are closely expressed here at dawn, are now being expressed at all different times of day. And that's true for many of these time points. Some of them are worse than others. We see some where you get really big distribution of the patterns and they're no longer expressed as a cohesive unit. If we looked at just the differentially expressed transcripts and we plot them by their peak in expression, so this is their peak in expression under normal nighttime temperature, this is their peak in expression under warm nighttime temperature. If they were the same, we'd expect this black line. And what we see instead is this white line. So it's higher, it's above the black line here and below the black line here. And so what this means is that here, these genes that normally peak early in the morning, these are those photosynthetic genes, are coming up later. They have a phase delay. Their peak expression is delayed. And these genes at night that are normally peaking, at ZT17 are now shutting off earlier, they're peaking earlier rather, under warm nighttime temperature. So they have a phase advance. So these are really two patterns we're seeing. And then we did a bunch of other analysis to show that this is circadian regulated. I'm not going to bore you guys that are not circadian aficionados with that, but to highlight these two groups. And so if we look at the genes that really make this criteria for being in these two patterns, what we see is those ones with the phase delay that are coming up later in the morning, those teenager genes, they're all photosynthetic genes. So really photosynthesis is delayed. And this is interesting because there's some debate about the effects on photosynthesis from the previous night's warm nighttime temperature. Some people say there's an effect, other groups say there isn't. And so from this data, assuming the transcripts, have some connection to the proteins and metabolites, which we have not shown that yet, I would say it probably depends on the time of day you do that analysis. Because if you do it here at these early time points, it'd be more likely to see a difference. And if you do it at the later time points when the warm nighttime temperature plants are trying to catch up, you might not capture that difference. And then the other group of genes that is really strongly is these ones that have the phase advance. So what we see here is that under normal conditions, the plants are continuing to increase until pretty late time point before dawn, and then they're going down. In contrast, the warmer nighttime temperatures are shutting off earlier. And these genes are enriched for genes that are involved in kinases, a lot of signaling components, phosphorylation pathways, and other signaling components. And so, we know that nighttime respiration is increased under warmer nighttime temperature. At this point in these sample collection, it's already acclimated. So these could be signaling pathways that could be feeding in to try and compensate for that increase in respiration. So these are really two big categories. So this is kind of generally looking at it, but what my lab really loves to do is to make gene regulatory networks and find candidate regulators. We're a transcription lab. We're interested in finding those hub genes, which could be targeted by breeders to then make plants more tolerant to warmer nighttime temperatures. So we wanted to make a transcriptional network. And the way we did this, and I won't go into super detail, but I'm happy to talk about it, is we took another data set that had somebody did this massive transcriptomic experiment in rice. They had over 500 field-grown samples. And so we built our gene regulatory network from that data, which has no warm nighttime temperature information in it at all. And so that by building the network, what I mean is we know, gene A regulates these genes, gene B regulates these genes, and these genes regulate gene A. So we made those connections from this data set. And then we did a couple things. We asked, of those regulators, which ones have targets that are really affected by warmer nighttime temperature, more than you would expect by chance? Which ones are enriched and have the most targets affected? Because those may be regulators that are really making that effect on the warmer nighttime temperature response. And then the other thing we ask is, how are those connections affected when we do the network under warmer nighttime temperature? So I'm just going to give two snippets for those. So if we look at the ones that have the biggest targets that are affected and we plot those out, so these are our top genes for having their regulators are disrupted under warmer nighttime temperature. They show differential expression. What's exciting to me is that most of these are clock associated. I'm particularly excited about this top one, BBX24. It's known to be involved in circadian regulation in Arabidopsis. And so we're excited about it as maybe a potential target for mediating these high nighttime temperature effects on this disrupted circadian rhythm that we see. And then, and several of these other top targets are also interesting. We're also interested in BDX24 because from the other analysis where we look at how those networks change, we see that it really is changing its targets as well. Not only are its targets disrupted, but it's changing its targets under warmer nighttime temperatures. So that's kind of interesting to us. Another analysis we did was we looked at the clock component. So this is just the clock regulators, the transcriptional regulators that are involved in the clock. And this is kind of a classic picture of them. So they're sorted by their time of expression. And this looks like, we did this from our data, and this looks like a classic rice clock picture. This is what they should be. So our data reflects chamber grown rice. It really mimics all the rice clocks that we know about. And that's good because this is indica, and most of those are done in japonica. So it's very consistent with that. So, however, if we compare that to the same network we built from the warm nighttime temperature data, we can see a lot of changes. So we see, at the dawn ones, we have more connections and more genes being expressed here. These 17.5-hour peaking ones are completely gone in warm nighttime temperatures. We don't have any in dusk here. We see a lot in dusk here. So really these connections for the clock are changing, which is of course consistent with this entire disruption of the rhythmic expression that we see. So this is, again, arguing that the clock is central. And so I'm not going to go into too much detail on how we validate it. Our intent was to validate it using the same ceramic heaters, but those got blown away by a typhoon. So Dr. Jagadish enabled us to find these heat tents. So we did our validation in these heat tents. And what was really cool about this was that we could do a gradient of nighttime temperatures. So we could do different nighttime temperatures and step them up. And so our validation is fairly strict. And we asked, for BBX24 and its targets that are affected, are its targets affected linearly in these validation experiments? And we saw that some of our key candidates like BBX24 and some of the PRRs were validated using this. And again, this is all published, so I'm kind of rushing through it to just get you the point and to make you excited to go look at that paper or to talk to me more about it. So the bottom line from this is there's a lot of research going on in rice and wheat and sorghum. Nighttime temperatures, which we are expecting to see, this is not, the 100-year progression, this is the 15-year progression, are going to have bad effects on rice yield and probably wheat yield and probably sorghum yield. Another thing I'm worried about is warm nighttime temperature improves the growth of some pathogens like botrytis. So we know botrytis grows faster under warmer nighttime temperatures. We've shown that warmer nighttime temperatures disrupt this fundamental coordination between the clock and the clock regulated genes, so the rhythmic gene expression is affected. We know that rhythmic gene expression is really important for biotic stress response, for abiotic stress response. And so now if we're disrupting that, the plants are unhealthier to begin with, the pathogens are happier, how is that intersection gonna really affect the plant? And if it's not gonna be able to mount the same response to pathogens, to drought stress, to vapor deficit changes, which are going on and probably causing some of these effects. And so, really this is kind of this very scary thing that's going on. And so that leads me to the next point, which is, climate change is really scary. When we hear about climate change on the news, when I hear about all these reports, I think they don't even know about nighttime temperature, and this research is really depressing. And we are not making the targets for hitting, the minimum climate change that's going to have them. We are still at the maximum carbon emission, so we're not getting there. And so I had essentially a mid-career panic and decided, so this also happened during COVID. And I also got tenure during this time. So this culmination of factors, I was like, I need to do something for my own sanity that's not just looking at the effects of climate change, but actively, proactively fighting climate change. So this is why I want to talk to you guys. I know I don't have a lot of time left, but I'm going to fit in a little bit on this project because I'm super excited about it. So I was having this panic and I was trying to keep myself positive and looking at all the cool things people were doing. So people were making roots bigger to sequester carbon. Ryan's doing this, carbon conversion. That's all really cool. But if we want to get rid of carbon, we have to go, to wind energy, renewable energy. That's where we have to be. It really needs to be there. And to get there, we need an electric grid. We need to be on an electric grid. That's really the best way to do it with our current technology. And really to get there, we need to have electrification. If we want a green economy, we need to be electric. That gives us more versatility to plug in solar, hydrothermal. Anyway, I'm not an economist, but that's, the bottom line. If we want to have electrification, we need these critical elements. And right now, to get these critical elements, we're destroying our environment. So this catch-22 kind of thing. So I saw this is kind of an opening. These are the critical elements we need, and highlighted in blue here are the rare earth elements. So scandium, yttrium, and the lanthanides here. And so these have important roles that we already use a lot of them for catalysis. If we want to move to energy independence, if we want to use to we want to have the neodymium magnets for wind turbines, we want to have light motors for light efficient vehicles, we need to be using these elements here. They also have roles in health and they have roles in defense as well, which is a good source for funding for these things. So the US uses a lot of rare earth elements, but we import 100% of these. And so that is a concern for this defense bucket here. They don't want to be importing these. And we import most of them from China, as does the rest of the world. It's not just us. Almost everybody that's using these imports them from China. We've been increasing our mining. So that's the purple here. And you can see this is 2018. We mined some. Why did you say in 2019 that we imported 100%? Because we don't have the processing capacity here. So what we do is we mine it, we ship it to China, we have them process it, we ship it back, which is not very environmentally friendly. And so, this is this kind of conundrum. And so my grad student told me about this movie, which I haven't seen yet, but maybe some of you have. And this really exemplifies this political nature. So I'll give you the plot of the movie. I haven't seen it, so I'm sorry if I ruin it. But the concept is there's a comet coming towards the Earth. It's full of rare earth elements. Do they destroy the comet now while it's far, far away and save the Earth? Or do they wait and take a chance and get closer and get those rare earth elements, but maybe risk not being able to divert it in time and destroy the Earth? So, okay, that's a made up scenario. It's a really obvious impact of comet. But what I'm arguing is that we're already doing that with our environmental destruction. So we want these rare earth elements. We're getting them using extensive carbon. Our mine in the US is in a really water limited area. It takes a lot of water to run these mixer settlers to separate them. It's very damaging. When we mine them, they're frequently co-occurring with radium and with radioactive materials, uranium and thorium. We don't do any recycling of them because it's very toxic to recycle them. And so, it's not a comet, but we are just, we're putting the Earth at risk for these rare earth elements. But at the same time, we really need these rare earth elements to have a green economy to save the Earth. So I felt like here's an open area and plants have a role. So what's the problem with rare earth elements? They tend to be really dispersed. You don't find them in veins like you do gold. You can find them in veins, but it's pretty rare. They tend to occur with these radioactive materials. And because of their valence chemistry, they're really difficult to separate. And so it requires a lot of energy, water, and carbon to separate them. But we have lots of rare earth elements in the US. So we have sufficient rare earth element sources, and I'm going to talk a little bit more about those. But plants are a solution to these. So what do plants, this is a plant breeding talk, what do plants have to do with the price of REEs? We use rare earth elements. Bacteria use rare earth elements. So there's rare earth element dependent requiring bacteria. Plants use rare earth elements, and plants use some of these higher rare earth elements. So they've been shown to be involved in some plant biochemistry and photosynthesis. And so plants are excellent at sensing ionic changes in their environment. They can hyperaccumulate rare earth elements, other metals as well. They can preferentially uptake some metals over others depending on the species. So they can do some of this pre-separation between the low and the high rare earth elements that we want. A lot of species don't take up the radioactive elements, and they're compared to these mixer settlers, they're very cheap to grow and operate. I know I'm talking to breeders here, you may think not cheap, but comparing this to these, mechanical processes, they're very cheap to grow and operate. So we have this project to try, and I'm not going to go through the whole project, but just to kind of get to the bottom line here, to really try and develop a phytomining system for developing rare earth elements. And so I'm just going to hit a few key points. With Mike Kudinoff in the College of Engineering, he developed this way to detect rare earth element uptake. So this is showing a pokeweed leaf. It's taking up cerium, and we can actually see the cerium accumulate in the plant. So now we can now detect which plants are in a high throughput manner, in a non-destructive manner, which plants are accumulating different levels of rare earth elements. So we can select for the trait we want fairly rapidly. We've done some selection of different plant species. We have a few candidates we're really interested in. I'm just going to highlight my favorite here, which is pokeweed or Phytolacca americana. It can accumulate up to 6 to 10% of its above ground dry weight in rare earth elements. This is not going to be realistic because we're just feeding it as much as it'll take up, and it'll take up as much as we give it. It loves rare earth elements. It's very tolerant to them. It actually pulls them in. And the other cool thing about pokeweed is that it preferentially, some varieties, some species, some individuals preferentially accumulate the valuable rare earth elements over the unvaluable. So this may not look like much of a difference, but 30% increase is actually fairly substantial for enriching for these elements. So it's already doing some of this preferential uptake. So, you know, we're not going to be putting pokeweed plants in this mine in California, but there's a lot of rare earth elements available in North Carolina in acid mine drainage and coal ash. So right now in the coal ash we produce from coal mine processing yearly, there's enough rare earth elements to supply the US needs for rare earth elements. The problem is it's really lower concentrations and it's not of interest to the mining companies because it's, you know, the investment, it's in dispersed areas, the investment's not worth it to set up the mining, you'd have to transport. But that's perfect for plants because there's not as much investment to transport plant seeds, and it isn't, you know, we can quickly develop it, and they're very dispersed. There's, you know, pools of fly ash all over North Carolina. We can take plants there. We can set up a processing plant for the plants nearby. It's not the same as bringing in 100 mixer settlers and the water and the electricity needed for that. So this is my pie in the sky plan, is to develop pokeweed. We have some candidate individuals that are really high at accumulating it. We have other species as well. We have duckweed species that are tolerant or sensitive. And so we want to develop these into systems to mine this waste for the rare earth elements. And the idea is to just use it, the money for that, to pay for cleaning up the waste. So we'll do two things at once, a win-win situation. The world's a wonderful place. We can clean up the fly ash ponds while we are getting the rare earth elements out. We're supporting a green technology, and I can go to sleep at night because at least I'm not doing something that's super depressing and I've got something to be happy about. So that is my pokeweed slide. I do have a lot of acknowledgements, but I'll go through them really quick because I know I'm over time. This is my lab right now. I am so lucky to just have amazing students who are wonderful and really great. In the lab right now, who's been working on the rare earth element projects, is Ed Moritz, and a wonderful undergraduate, Alex. Previously, Dimitri, Kevin, and Jigger worked on the nighttime temperature tolerance I talked about, the time of day. Jigger did all of the warm nighttime temperature stuff by himself. Stephanie was a technician, Faith was an undergrad, and Cyprian was a postdoc that have worked on the rare earth element mining. I have really great collaborators. Krishna, all the warm nighttime temperature stuff is all with him. We have a great collaboration with a company here in the Triangle for developing new methods for analyzing gene regulatory networks. Mike Kudinov has really been instrumental. He's been the leader in this detection of rare earth elements, and we've been trying to work with other people to advance this. And also Amy Grundon for some microbe work in the rare earth elements that I didn't talk about. A little bit over, I'm sorry, but, and I know probably too fast for some people to understand, but I wanted to get the rare earth elements in.
Thank you so much, Colleen. I love the passion that you put in your work, and I got really impressed the first time I listened to your night expression differences, and because I commented to you, it reminded me of when I was a popcorn breeder. The key element was night temperature, and I thought, and then in wheat the same. We were measuring daylight canopy temperature, and I think we should have been measuring night temperature. Now with the devices we have, we could do that. So we have okay, we can entertain a few questions. Can we open the microphone for Brandon, for Jim to you have a very long question there. I don't know if you can read it, Colleen.
I'm trying. I'm trying.
Jim, can you speak?
I'll let Colleen read, and then I'll try to restate it. This is the breeder's question, Colleen.
Yes.
Okay.
So you knew this question was coming, which is, it's clear that you've identified in really good detail how the increased nighttime temperatures are affecting these regulatory pathways. And then my question is, okay, so let's say I'm a rice breeder at Erie, and now I've seen your paper. What's the action plan? Like, how do I take that information and turn that into, improving tolerance to the increasing nighttime temperatures?
So one of the ways we're doing it is that there are a number of QTL analysis for warm nighttime temperatures going on, and what we're hoping is that these regulatory networks might identify candidate genes within those regions, or narrow down the region, or help with that. So that's one, easy answer that I can say we're doing right now, is to try and connect this data with QTL data to try and see if that matches up. I mean, who knows if it will, but that's one of the steps we're taking. If we validate these, which we haven't yet, but for example, if we validate that BBX24 is a regulator of these, genes, I think it would be interesting to try things like, and this is the molecular person talking and totally not a breeder talking, so, but can we select for, can we use that as a marker for breeding? Not necessarily selecting on that gene, but can we use that, like watching the regulation? So one of the problems with warm nighttime temperature and a lot of other stresses is that if we were to do a very large breeding program for it and, heat up the field, we would be causing climate change. It's kind of these like problems. You don't want to like actually do the experiment to select for the plants, because the electricity we would use would be kind of counterproductive and expensive. And so could we do it on a smaller scale, look for plants that have more variation in that? This is, again, I'm not a breeder, maybe this won't work. Plants that have more variation in that gene, and then use those as a starting population to look through smaller set and heat those up with nighttime temperature. I don't know if either of those would work. Those are just ideas.
Thanks.
Thank you, Jim, for your question. I thought about that too, and most of your results are sort of unidimensional in a sense. It's a genotype. I wonder how much diversity it is. That's the other question.
Yeah, so there is, so there are QTLs that have been done, to look at these, and then there's a few things out there. The main problem is that the warm nighttime temperature in the greenhouse or glass house or chambers doesn't really replicate the warm nighttime temperature in the field, probably because of the vapor pressure deficit. Or it could be the root versus shoot temperature difference. So there is a lot of variation genetically in rice, and then there's variation. So there's a group doing this in wheat, also doing the QTL analysis for high nighttime temperature tolerance.
So the last question before we close the seminar, and you can always contact Colleen for follow-up questions, of course.
Please do, especially if you've got ideas for breeding or
so
how to
is there a
with.
good collection of pokweed genotypes to search for more efficient variants?
This is where I need to talk to somebody, because we don't even know if pokeweed selfs. We know that pokeweed we collect from different areas has different, if we do like rapid DNA testing, we can see different DNA, we can see different markers, and we can see different uptake. And we can clonally propagate it. So we have we have these super high uptake, we can clonally propagate them, we have those, but we don't know what to do next. How do we select for ones? What do we do next for this kind of stuff? So really need to talk to somebody.
That's one of the reasons why I brought you the relative of the Phytolacca dioica, which is male and female. That the sexes are separated.
Yeah, so we have 4 species of Phytolacca, and some of them do have different sexes.
Okay, I'm gonna share the screen here to talk about next week's seminar. Thank you so much, Colleen. Very, very good seminar. A lot of ideas there to munch on. Next week at the same time, we're going to have Amanda Holsekamp from NC State USDA talking about interdisciplinary collaboration to build genomic support capacity for public cotton breeding programs. Thank you so much. The recording for this today's seminar is going to be shared together with the announcement for Amanda's seminar probably tomorrow. So if you guys missed some part of the seminar, can always catch it up through the recordings. Thanks, Brandon, for your help today. And I appreciate your attendance. Have a good rest of the week, a good weekend, and I see you all next week at 11 on Thursday. Thank you so much, Colleen, okay?
Thank you. Thank you so much for having me.
Okay, bye now.